Hello everyone. It is actually 4 o'clock in the morning right now, and so I am still pumped and ready to go and ready to show you guys another virtual lab tutorial on meiosis, which is the process of synthesizing sex cells like sperm and egg cells. And so the purpose of this video is to just show those students who already have a background in meiosis how to draw those phase diagrams with chromosomes, spindle fibers, centrioles, DNA molecules, etc., etc. So this is what this lab is for. It's not to teach you the concepts of meiosis, it's just to teach you how to draw the diagrams of meiosis. And these two things go hand in hand. So once you know the concepts and once you know how to draw the diagram, then it starts to make perfect sense for you guys. So without further ado, let's go ahead and go to the whiteboard and find out how to draw meiosis. Now I recommend before we get started that you take out a blank sheet of paper and you follow along with me as I'm drawing, because one of the best things that my students in the past have told me is that when they draw these diagrams along with me whenever I'm doing it on the white or the blackboard in a classroom, it really solidifies mitosis and meiosis. In this tutorial, it's going to be meiosis. So let's go ahead and get started right away. All right, so now we're going to go ahead and start talking about meiosis. And this is a virtual lab. I like to call it a virtual lab. But pretty much what I'm going to do is I'm going to be drawing each of the phases of meiosis. I'm going to kind of describe it a little bit and then count the number of chromosomes, chromatids, and DNA. And hopefully from this exercise you'll be able to do it on your own as well. One of the big things that I recommend that you do is as I'm drawing this, you should also be drawing along with me. That's one of the big key things that so many of my students have told me whenever I'm drawing this on the white or blackboard. When students follow along, it kind of solidifies it in their mind just to kind of see it and walk through this drawing with their instructor, with me, pretty much. So let's go ahead and get started with this lab, virtual lab. So the learning objectives are these: knowing how to draw all the phases of meiosis, drawing each diagram of meiosis with the correct number of chromosomes, chromatids, and DNA molecules, and also being able to draw all of the relevant structures. Now, you should already have a basic understanding of meiosis. If you don't have this basic understanding yet, I recommend that you check out some of the online YouTube videos, or maybe discuss this with your instructor, or read up about it, catch up about it in your biology textbook. Okay, but this lab, this virtual lab, is just strictly for students who want to know how to draw each of those phases of meiosis. That's it. Okay, so let's go ahead and get started. All right, first of all, we're going to have to learn how to count the number of chromosomes, chromatids, and DNA molecules. All right, now for this, I like to use an abbreviation for chromosomes. I like to use CH' for chromosomes, CH' for chromatids. Okay, it just makes it easier for us, so I don't have to write too many letters on the small whiteboard that I have here. So let's go ahead and get started with this exercise. Here. All right. So let's take a look at this example. In this example, I have 2 DNA strands, and so therefore 2 DNA molecules. Okay, I'm just going to say 2 DNA. I also want to know how many chromosomes do I have. A chromosome, in my class, I like to describe a chromosome as like an entity of DNA, one solid piece of DNA. I see 2 separate entities of DNA here, 1 and 2. So I'm going to say that there are 2 chromosomes in this diagram. How many chromatids do we have? In order for it to be a chromatid, they have to be connected at something called the centromere. But we don't have a centromere here. These are 2 separate entities. So therefore we have only 0 chromatids. Let's do another example. How about this? In this example, I have 1, 2, 3, 4 DNA molecules. Okay? Now, how many chromosomes do I have? Well, 1, 2, 3, 4. There's still 4 separate entities. So therefore, there are 4 chromosomes. How many chromatids do we have? Well, they're not connected at the centromere, so therefore we don't have any chromatids in this example. All right, let's try another example. All right, how many DNA molecules do we have? Well, I see 1, 2, 3, 4. So 1, 2, 3, 4. There's 4 DNA. How many chromosomes do we have? Well, there's one piece here. That's 1 chromosome because it's one solid piece. Well, it's not solid, but I like to think of it as a solid. Another one here and another one there. So we have 3 chromosomes. And I'm labeling these in blue so you can see the difference. Now, how many chromatids do we have? Well, in order for it to be a chromatid, it's got to be connected at that centromere. Okay? So we have here 1, 2, but that's not a chromatid because it's not connected at a centromere, and that's not one either. So we only have 2 chromatids. All right, let's do one more example here. All right. So how many DNA molecules do we have? Well, we have here 1, 2, 3, 4. It's still 4 DNA because we have 4 strands. How many chromosomes do we have? Well, here's one piece, and here's one other entity, so I only have 2. And lastly, how many chromatids do we have? We got to have them. Chromatids are there when there's a centromere. There's a centromere here and here because they're connected together. So we have 1, 2, 3, 4 chromatids. All right, hopefully this exercise, or this sample exercise here, is kind of a good way to show you how to count the number of chromosomes, chromatids, and DNA molecules, because this becomes extremely important in the next segment when we begin to count those. All right, so let's go ahead and move on to meiosis. All right, let's talk meiosis. Now, in meiosis, we have these following phases. We can break them down into meiosis 1, and we have here meiosis 2. Okay? First step here is prophase 1, metaphase 1, anaphase 1, telophase 1, and there's also cytokinesis here too, but I'm not going to worry too much about that. All right, when we move on to meiosis 2, we have here prophase 2, metaphase 2, anaphase 2, and telophase 2. All right, and meiosis 1 happens, and then meiosis 2 happens after that. Okay, I have a small board, And I need to draw 8 of these different diagrams in this tiny little board. So we're going to have to do some abbreviations here. So just bear with me. When we talk about prophase 1, I'm going to label that as P1. Metaphase 1, M1, A1, and T1. Okay, so that you know. I'll put a little dash there too, so you know. Same thing here with the meiosis 2. P2, M2, A2, and T2. All right. So these are the abbreviations that I'm going to use in the diagram. All right, so the moment you've been waiting for, let's draw each of those cells in meiosis. Okay. All right, so let's go ahead and start drawing those diagrams in meiosis. Now, before we begin, I'm going to tell you that in this exercise, or in this virtual lab, there are 4 chromosomes. So the number of chromosomes is equal to 4. Now, some instructors, they like to write it like this. 2n is equal to 4. Doesn't matter. It's the same thing. Now, some of you may also know that there is a phase that's called interphase, where we have G1, S, and G2. Okay, that's interphase, but we're not going to worry about that in this lab. We're just going to draw the meiosis 1 and meiosis 2 and get those diagrams out of the way. All right, let's go ahead and move on and get started with that right away. We have here meiosis 1. This is the first part of meiosis. Here's our cell. We have this solid circle. And inside of this circle we have a dashed inner circle, which resembles a degrading or a nuclear envelope that's breaking down. This is again prophase 1. We have our star-like structures and these little projections that are sticking out of it. They kind of look like a shooting star, and there's 2 of them. There's a pair of them. The star portion is called the centriole, and the tail portion with those little projections is called the spindle fiber. All right. Okay, now this is very important. Okay, this is extremely important that you watch how I draw this. Okay, watch how I draw these chromosomes. Extremely important. This is what makes it different from mitosis. If you watch my mitosis video, this is what makes it different. Watch how I do it. There's 1 chromosome, and look what I do. There's a second one. Here's our 3rd one, and here's our 4th one. Notice how they're kind of lined up differently. Okay, we have a little touching here, and we also have another touching here. This is called crossing over. Okay, and this is key that you understand that crossing over happens in prophase 1. You have to understand that. Okay, crossing over happens in prophase 1, and this gives the chromosomes and the genes and the genome some genetic diversity here and some uniqueness. Okay? All right. So let's go ahead and label the rest of our diagram here. We have the nuclear envelope. Okay? And of course, crossing over. Let me go ahead and kind of label it there. All right. What's next after prophase 1? Well, you probably guessed it. It is metaphase 1. Here we go. Metaphase 1. Now we have an imaginary line down that middle, right here. Okay, and this is called the metaphase plate. Okay, it's called the metaphase plate or equatorial line. All right, that should be an E. Okay, you guys have to watch this too. Watch how I do this. Okay, this is extremely important. If you watch my mitosis video, you're going to find it's different in meiosis. Okay, so watch how I do this. There's one, another one. There we go. There we go. Notice that the homologous chromosomes are lined up. These are called homologous chromosomes. So I like to think of homologous chromosomes as this. You have a deck of cards, and one deck of cards has the Queen of Hearts, and the other deck of cards, say you have a second one, that deck of cards also has a Queen of Hearts. So the Queen of Hearts from the first deck and the Queen of Hearts from the second deck are called homologous chromosomes in this case. Okay? Your mom gave you 23 chromosomes and your dad gave you 23 chromosomes. So it's like you having two decks of cards in each one of your cells. Okay? Except each deck has only 23 cards instead of 52. Hopefully that makes more sense. All right, let's continue on with our drawing. We have here our centrioles, and they have already migrated to opposite poles. And then we have our spindle fibers that are coming out and projecting on out, and they are touching that centromere on each of the two homologous chromosomes. Same thing here. At the lower pair, we have that going on. Okay? And then, of course, we have a non-kinetochore, non-connecting kinetochore spindle fibers, but I'm not going to worry too much about that. Okay. All right. What's after meiosis? What's after metaphase 1? We have anaphase 1. Okay. And in this case, the homologous chromosomes separate, and that's key. I wouldn't be surprised if maybe your instructor asked you on a test question what separates in anaphase 1, and you would have to say homologous chromosomes separate in anaphase 1. Okay, so we have here our centrioles. They're coming out, they're touching the centromeres of each of the homologous chromosomes there, and they're being dragged across to opposite poles. Okay, I would definitely want to write that in my notes. Homologous chromosomes separate. Okay, I would definitely write that in my notes. And then lastly, for meiosis 1, okay, we have there the splitting of that cell. We have the nuclear envelope that's coming back. Okay, and we have our homologous chromosomes are now in each of those nuclei. Okay, and we have something called the cleavage furrow. First, and then now we have our centrioles, our spindle fibers are breaking down. They're no longer needed. This is telophase 1. All right. But this is just meiosis 1. We need to get to meiosis 2. We're not done yet. Okay? And now I'm just going to take one of these cells. We're not going to do two, just one of these cells. So I'm going to take one right here. I'm going to call this obviously prophase 2. That's the next step. Right, so we're only doing one of these. Okay, so that's one of these. Now notice this. If I'm doing one of these, you always want to make sure that you understand that there are actually two of these cells that are going through meiosis 2. So I like to put a times 2 at the top to remind me that there are actually two of them, but this is only one. Okay? All right. So we have here, oops, I'm getting old. We have here our nuclear envelope is breaking down. Again, we have our centrioles and spindle fibers. We have here our chromosomes. What's after prophase 1? We have here metaphase 1. Metaphase 1. We still have that imaginary line coming up in metaphase 1. Now this is what is different compared to metaphase 1. Sorry guys, that's metaphase 2. Alright, watch this guys. It's the sister chromatids this time that line up at the metaphase plate. Okay? Sister chromatids line up at the metaphase plate. Okay, there's our centrioles. They're touching. And of course we do have non-connecting kinetochore spindle fibers, but I'm not too worried about that. Okay. What's after metaphase 2? We have anaphase 2. Anaphase 2. And in this case, the sister chromatids separate. Watch that. Okay, that sister chromatid separates. Okay. We still have those non-connecting spindle fibers. And then lastly, we have here telophase 2. We have our nuclear envelope that's coming back. Now we have this. Centrioles are there. Spindle fibers are breaking down, and we have once again our cleavage furrow breaking these in again. Now remember, there's two of each. Okay, there's two of those. So we start off with one cell, and we should end up with 4 cells at the end. 2 here times 2 is 4 down here. Okay, and that's telophase 2. All right. Major note here, sister chromatids line up. Okay, at the metaphase plate. In anaphase 2, sister chromatids separate. Okay, versus in anaphase 1, homologous chromosomes separate. In metaphase 1, homologous chromosomes line up at metaphase plate. Okay? Should say plate. All right. So now you have all the diagrams, but let's count the number of DNA, chromosome, and chromatids in here. So I'm going to go ahead and use my orange marker here. I'm going to say the number, well, this orange marker is terrible. Let's go ahead and just use red. Number of DNA, number of chromosomes, number of chromatids. All right. So how many DNA molecules do you see in prophase 1? I see 1, 2, 3, 4, 5, 6, 7, 8. Okay? How many chromosomes? I see 1, 2, 3, 4. Now, why is it 4? Some of you guys have watched my mitosis video and you heard that, well, if they are touching, they should be considered one chromosome. If they're one entity, well, they're not here and crossing over. They're not actually touching. They're just hovering over each other. They're not actually attached or stuck together like at the centromere. So therefore, we still consider these 2 separate chromosomes. And in addition to that, for some of you guys, there's an old-fashioned term to describe this coupling here. This, you might hear it being called a tetrad. Okay? Just so that you guys know. But we still have 4 chromosomes total. There's 1, 2, 3, 4. And how about the number of chromatids? Well, we have 1, 2, 3, 4 centromeres. And if you multiply that by 2, you'll get 8 chromatids. Okay? Or just count them. There's 1, 2, 3, 4, 5, 6, 7, 8. How about here in metaphase 1? Well, the number of DNA molecules we have here, 1, 2, 3, 4, 5, 6, 7, 8. So we have 8 DNA molecules. How about the chromosomes? Same thing. 1, 2, 3, 4 chromosomes, just like before. And chromatids, same thing. It's still 8. Okay? How about here in anaphase 1? Well, how many DNA molecules? Well, 1, 2, 3, 4, 5, 6, 7, 8. We still have 8. How about chromosomes? I still see 4 entities. And how about chromatids? Well, 1, 2, 3, 4, 5. We still have 8. Okay? Now, how about in telophase 1? Remember, it's per nucleus. Okay? So in telophase 1, I see 1, 2, 3, 4 DNA molecules. I also see 2 chromosomes. There's 2 entities here. And I see 4 chromatids, or sister chromatids. All right, let's get over here to prophase 2. Well, how many DNA molecules do you see? Well, I see 1, 2, 3, 4. How many chromosomes? Well, I see 2 entities. And how about how many chromatids? Well, got 2 centromeres, so 4 chromatids. 1, 2, 3, 4. How about in metaphase 2? Okay, well, I see 4 DNA molecules. I also see 2 chromosomes, 1, 2, and I see 4 chromatids, 1, 2, 3, 4, because we have a centromere here. All right, how about anaphase 2? This is a tricky one, okay? How many DNA molecules do you see? Well, 1, 2, 3, 4. How many chromosomes do you see? Ah, we have 4 chromosomes here. Okay, I'm going to put a little star there to kind of bring your eye there. We've got 4 chromosomes here because we have 4 entities. And how about how many chromatids do we have? Well, we don't have any more centromeres per se. We have 0. They're technically still called centromeres, but we don't have any connecting centromeres where they connect with the other sister chromatid. So we don't have that, so it's still 0. Okay, it's now 0, I mean. Now, how about in telophase 2? Well, how many DNA molecules do we have per nucleus? Well, we have 2. How many chromosomes do we have? 2. How many chromatids do we have? None. Okay. Amazing. All right. Now, just a side note for you guys. In telophase, the chromosomes are supposed to be uncondensed. Okay? Chromosomes are supposed to be uncondensed. Let's move that up a little bit. Okay. The reason why is because at that point in time, the chromosomes are no longer needed to be packaged. They finally hit their destination. They're finally in their daughter cells, and so so the DNA begins to unravel. There's no need to be tightly packed anymore. Okay, and so just keep that in mind. In my drawing, I still kept the chromosomes condensed because when I'm grading these, I like to make sure that the students understand the total number of chromosomes, chromatids, and DNA molecules. But just know this, that if you're in my class, I ask you to keep the chromosomes condensed so I can count them when I'm grading, but then I want you to make a note here that they're supposed to be uncondensed. They're supposed to be unraveled. Okay. All right, so hopefully this helps explain how to draw meiosis and all of the different phases of meiosis and what's relevant in those phases. And don't forget, the purpose of meiosis is to create sex cells like sperm and egg cells. And so you start off with one cell here, you end up with 4 over there. Okay. All right. Hopefully that you found this video useful and helpful. Now you're able to take any number of chromosomes, change this number, and do this diagram on your own as well. If you're in my class, you should be able to draw this diagram like the back of your hand in less than 5 minutes, and being able to do all of these numbers and calculate the number of chromosomes, chromatids, and DNA molecules. You should be able to do that. All right. So anyway, hope you found this useful. All right, so I hope that you guys found that meiosis video very helpful. You guys, if you think about it, you just synthesized 4 gametes, okay? And those gametes can either be sperm or egg cells. So you either made 4 sperm cells or you made 4 egg cells, okay? So which one did you make? It's up to you, all right? But anyway, hopefully you guys found that helpful. If you didn't quite understand it, I strongly recommend that you rewind this video, move that slider back, or whichever direction it's supposed to be, and move it back there and follow along with me step by step. Use colored pencils if you have to, so that you can kind of see what's going on in those diagrams. And the thing is that once you know these diagrams, it's going to make sense. The concepts of meiosis along with the diagrams go hand in hand together. It's going to make perfect sense for you. Every time I have taught mitosis or meiosis, in this tutorial is meiosis, every time I've taught it, students have always understood it straight out, because whenever I draw it on the white or the blackboard, they totally get it from there. So hopefully you guys followed along with me, sorry, tongue twister there, that you guys followed along with me and are now beginning to understand the process of meiosis. Okay, well anyway, hopefully that helped you guys, and without further ado, I guess that's sayonara. All right, well anyway, see you around the virtual world, guys.